3. Biochemistry of cell. You all know that cell is a fundamental, structural and functional unit of an organism. The living cell is having the ability to carry out metabolic activities, and for this we have to understand the chemistry of various organic molecules found in cells. Metabolism means a variety of reactions carried out at cellular level. The collection of various types of molecules in a cell is known as cellular pool. It consists of inorganic materials and organic compounds. The metabolic reactions are divided into two main categories. First one, anabolic reactions. Here, the biosynthesis of a new cellular material takes place. And catabolic reaction. Here, a complex storage product is hydrolyzed and broken down into smaller and simpler molecules. Both the reactions are catalyzed by biological catalysts called enzymes. Now this is the classification of basic chemical constituents. 1. Carbohydrates containing carbon, hydrogen and oxygen atoms. And the proportion of hydrogen to oxygen is 2:1. 2. Proteins consist of carbon, hydrogen, oxygen and nitrogen. 3. Lipids contains carbon, hydrogen and oxygen, but the proportion of hydrogen is to oxygen is greater than 2:1. Now first, carbohydrates. The simple carbohydrates are having the formula CnH2nOn and the complex carbohydrates are having the formula C6H10O5N. Now the first subclass, monosaccharides. These are the compounds which cannot be further hydrolyzed into smaller molecules. They are further classified as aldoses containing aldehyde group and ketoses containing ketone as a functional group. The number of carbon atoms are 3 to 6. They are crystalline, soluble in water and sweet to taste. For example, glucose, ribulose, etc. Then disaccharides. These are made up of two monosaccharide units and they can be hydrolyzed into monosaccharides. These are soluble in water, sweet to taste and crystalline. The covalent bond that joins monosaccharide units is called glycosidic bond. For example, sucrose, lactose. Then polysaccharides. A single polysaccharide may consist of thousands of units of monosaccharides. They can be easily hydrolyzed. These are amorphous, tasteless and insoluble in water. They are further classified as homopolysaccharide containing one type of monosaccharides or heteropolysaccharide containing different types of monosaccharides. For example, cellulose, starch. Then functions of carbohydrates. To provide energy for metabolism. 2. A monosaccharide, glucose, is the main substrate for synthesis of ATP. 3. In mammals, disaccharide lactose is present in milk which provides energy to their babies. The polysaccharides are structural components of cell membrane and cell wall and also serve as reserved food material. Then proteins. These are long-chain polymers of amino acids. They are macromolecules of high molecular weight. Some protein molecules contain sulfur and other elements in addition. In a long chain of amino acids forming a protein, the amino group, that is NH2, of one amino acid is linked to the carboxyl group of the other amino acid. The two amino acids are condensed by removal of a water molecule to form a peptide linkage. The remainder of each amino acid after removal of a water molecule is called residue. A molecule of a protein made up of two amino acid residues is dipeptide, of three residues tripeptide, and of many residues polypeptide. Now these are the some amino acids which occur in protein: glycine, alanine, cysteine, aspartic acid, glutamic acid, valine, threonine, etc. A protein molecule may consist of additional special bonds called disulfide bonds. Now, classification of proteins. Simple proteins, these are composed of only amino acid or their derivatives. For example, histone. Then conjugated proteins, these are simple proteins with some non-protein part called prosthetic group. For example, lipoprotein, that is proteins plus lipid. Nucleoprotein, proteins plus nucleic acid. Glycoproteins, proteins plus carbohydrates. Chromoprotein, proteins plus pigment. Now the functions of proteins. Most of the enzymes are proteins. For example, amylase. Hormones like insulin, growth hormones are proteins. Structural proteins form part of cells or tissues. For example, keratin in hair and skin. Then contractile proteins occur in muscles. For example, myosin. Transport proteins are useful for transportation of certain materials. For example, hemoglobin in blood for transport of oxygen. Then defensive proteins are useful for protection of body against diseases. For example, thrombin for blood clotting. Then lipids. The lipids contain two types of monomers such as fatty acids and long chain alcohols, commonly glycerol. Then classification. First one, simple lipids. These are the esters of fatty acids with alcohol. Depending upon the number of fatty acid molecules attached to the glycerol molecule, the esters are called mono, di or triglycerides. These are further saturated fatty acids and unsaturated fatty acids. Then compound lipids. These contain some additional elements or groups in addition to fatty acids and alcohol such as nitrogen, phosphorus, sulfur, etc. For example, phospholipids. Then derived lipids are the hydrolytic products of lipids. These are steroids such as cholesterol, waxes such as plant waxes, and carotenoids such as alpha and beta carotene. Function. Lipids act as high energy reserve food material. They are important components of cell membrane of eukaryotes. Lipids act as components of some enzyme system. Lipids act as insulator for heat. A lipid, cholesterol, takes part in the synthesis of vitamin D. Then nucleic acid. The nucleus consists of two types of nucleic acids, DNA, that is deoxyribose nucleic acid, and RNA, that is ribose nucleic acid. These are macromolecules composed of many small units or monomers called nucleotides. Each nucleotide is formed of three components, that is sugar, a nitrogenous base, and a phosphate. And when it is a nucleoside, it contains sugar plus nitrogenous base. Now the different components of nucleic acid are 1. Sugar. It is a pentose 5C sugar. Then nitrogenous bases. These are either purines, which are double ring compounds further distinguished into adenine and guanine, while it may be pyrimidine bases, which are single ring compounds further distinguished into thymine, cytosine and uracil. Then phosphoric acid. Phosphate is present in nucleic acid. Now structure of DNA. It is a double-stranded helix in which each strand is made up of thousands of deoxyribose nucleotides. They are linked with each other by phosphodiester bonds. The two strands of DNA are antiparallel, complementary and adjoined by weak hydrogen bonds. The N bases are complementary, that is, adenine always pairs with thymine and guanine pairs with cytosine. The DNA is the genetic material and contains all the information needed for development and existence of an organism. The quantity of DNA in each diploid cell is constant. Now the structure of RNA. It is single-stranded and the strand may be straight or variously folded upon itself. The nitrogenous base thiamine is replaced by uracil in RNA. The first RNA is genetic RNA and second one non-genetic RNA. Non-genetic is having three types: mRNA, that is messenger RNA, which carries the genetic information from nucleus to the site of protein synthesis. Then rRNA, that is ribosomal RNA. It is associated with ribosome. Then tRNA, that is transfer RNA. It carries activated amino acids to ribosomes and helps in elongation of polypeptide chain during the process of translation. Then enzymes. In living organisms, many metabolic processes occur at every moment in each cell. These reactions occur at normal body temperature and pressure due to the presence of specialized macromolecular proteinaceous substances called enzymes. These are synthesized by living cells. These are biocatalysts, which alter the rate of biochemical reactions without getting used themselves. The enzymes that are produced within the cell are endoenzymes, and those which act away from the site of synthesis are exoenzymes. Most of the enzymes are proteins. Now types of enzymes. 1. Oxidoreductases. These catalyze oxidation reduction. Then transferases. These help in transfer of chemical group between a pair of substrate. Then hydrolases. These catalyze the hydrolysis of ester, ether, etc. Then lysases. These catalyze non-hydrolytic cleavages. Then isomerases. It catalyzes interconversions of geometric, optical and positional isomers. Then ligases. It helps in joining the two molecules. Properties of enzymes. Enzymes accelerate the reaction but do not initiate it. Enzymes themselves do not participate in the reaction. The molecules of an enzyme is larger than that of substrate molecule, and hence during reaction a specific part of enzyme molecule comes in contact with the substrate molecule, and this part is called active site of enzyme. Then most of the enzymes are proteins which show the amphoteric nature. Most of the enzymes are specific in their action. The enzymes are colloidal in nature and they provide large surface area for reaction to take place. The enzymes generally work best under certain narrowly defined conditions called as optima, which include appropriate temperature and pH. Then inhibitors. The enzymes are sensitive to the presence of specific chemicals that bind to the enzymes. When the binding of the chemical shuts off enzyme activity, the chemical is inhibitor. The inhibitors that closely resemble the substrate in its molecular structure is called competitive inhibitor. Poisons like cyanide do not bind with enzyme at substrate binding site but bind at some other site and inhibit the activity of an enzyme, and such substances are called non-competitive inhibitors. Then cofactor. To make an enzyme catalytically active, it needs some non-protein constituents called cofactors. The prosthetic groups are those cofactors. Then concept of metabolism. The sum total of all the processes taking place in a living cell is called metabolism. Among these, the synthetic processes are called anabolic processes like production, and all the degrading processes of the cell are catabolic processes. Anabolic processes consume energy while catabolic pathways lead to release of energy, and this energy liberated is stored in the bonds of ATP.